initially envisioned for the Sampson lab is creating a team that was passionate in their hearts and their heads about finding answers for treatments and cures of nephrotic syndrome. I thought that if we could bring together folks with expertise in computer science, statistics, clinical nephrology, and biology, then we'd be able to work together to provide answers for our patients, for doctors taking care of them, and other people in the community.
I went to school at the University of Michigan School of Public Health for biostatistics, and I was funded by the Genome Science Training Program. So shortly after graduating, I wanted to do a job where I could apply what I learned in the graduate program specifically to a disease. And that's where I got linked up with Matt, where I learned about nephrotic syndrome and how I could use the statistical knowledge I had to try to understand genetic drivers of diseases. So throughout my time in the Sampson lab, I've worked on genome-wide association studies, or GWAS, where I looked for genetic variants that are associated with risk of disease. I've also worked on EQTL analyses, where I look to see if there's genetic variants that are associated with gene expression levels. I've also worked on studying the APOL1 gene and trying to compare FSGS patients that have this high-risk genotype versus those who do not. And I'm currently using single-cell RNA-seq data to identify cellular differences between people with and without the APOL1 genotype.
So my lab is specifically interested in how the transcriptional regulation in different types of cells contribute to the progression and development of the diseases, specifically focusing on kidney diseases. But that's the general. In order to understand that, we develop a lot of computational tools to help researchers to figure out and understand the gene regulatory network and how the non-coding element of the genome contribute and regulate the target gene's expression. In order to really demonstrate and then apply the idea that I have, I really need a solid and concrete example, such as kidney disease. After talking to Matt, I was really convinced that kidney disease is one of the ideal problems that we can tackle with the ideas that I have. Kidney consists of many different cell types, and they have a very complex structure that makes it ideal tissue for single cell study, which I'm very much interested in. There are multiple reasons why I chose to study kidney, but primarily it's because it basically gives me all kinds of opportunities to apply to my specialties and expertise. The main difference between our work and Sampson Lab is probably the focus and the specialties that we have. We are more specialized in computational bioinformatics and a little bit of less knowledge about nephrology, I would say. But we have a lot of experience, at least myself have a lot of experience in developing computational tools, bioinformatics tools over the last 15 years. And that basically contribute to our teamwork, and I think it creates a great synergy between them. Plus, Matt is nephrologist, and then there are several doctors in his group who really have expertise in nephrology and kidney diseases. If anything comes up that we don't really understand, they probably have better answers than us. If our work is more tailored toward the developing method, while Matt's group is more focusing on analysis side and the interpretation of the analysis. That's the main difference between the two teams.
It happens a little while ago as internal medicine resident when I am looking for places to do my renal fellowship. I was looking for places that can mentor me and give me legitimate aspect of precision nephrology that is applicable to the patient care. And at that time when I met Dr. Sampson, he's renowned in both the Mendelian and non-Mendelian genetic worlds, and as I've ventured into this precision nephrology, the best thing that I can do at that time is to start learning genetics and get mentored through the way to actually be able to apply this to patient care. So from that meeting in the Kidney Week, I decided to join the lab. The culture of the lab, I would say we are very open to ideas and to feedback and to criticism in the work that we do. So it really reflects in our lab meeting where we would share about our day-to-day work around once a week to keep people at least the ground word of what they are working on. I felt our lab meeting is a safe space to talk about everything, and we got a lot of good feedback from people that work around and share about our project freely, and that's very important for scientific inquiries and discoveries. My project specifically want to look at the impact of genetic variants in adults with nephrotic syndrome, which is the area that is not heavily studied in the past. We usually use genetic changes to take care of patients with full-blown genetic syndrome, that we look at them and we felt like they have a lot of things wrong in the body and that might be from a genetic change. But however, nowadays we know that these genetic changes may not be as big of an impact for everything in the body, but they can have some impact to specific organs. By knowing these things in the adult population, we should be able to incorporate the genetic analysis into the care of our patients with a lot of condition. And the condition I specifically study is the nephrotic syndrome in adults.
When I was coming to Boston Children's to be a fellow, I got really excited about the opportunity to use large data sets, computational approaches to try and better understand why kids were getting nephrotic syndrome. I knew that there had been a lot of research done here on a patient-by-patient basis to discover the cause of nephrotic syndrome, and a lot of good work had come out of that. And every field has a evolution, and it seemed like a lot of the individual results that could be found patient by patient had been found, and now is a chance to use a different type of data and a different approach to keep pushing these discoveries forward. I think that doing research on a regular basis keeps me really in touch with all the work that's being done to better understand this disease and better understand how we take care of these patients. And I think I have the chance to come back to the patients I see every day and think carefully about what makes their disease unique, what new approach can we use for them. And I think that's more important than the excitement and joy I get out of being in the lab and running an analysis and seeing an output. So I work on genetic risks for nephrotic syndrome. We now have really large collections of patients, hundreds of thousands of patients, which allow us to detect the impact of rare variants when they might not have such a significant effect. A lot of prior work in this field has found genetic defects which cause you to go from having no disease to having a really severe form of disease in almost every patient with it. But there's a lot of people who have variants in their genetic code that it doesn't take their risk from no disease to a 1 in a million disease. It takes their risk from normal amount of risk to double the risk or 3 times the risk. And these large biobanks allow us to understand those risks better, and those risks are applicable to a lot more people walking around every day.
Okay. So the BICID study is a biobank to eliminate the genomics of kidney disease, and it's basically a biobank that collects samples and patients' information, and we study the cause of nephrotic syndrome. My role in the BICID study is I am the coordinator, so I screen for patients that are eligible for the study here in the hospital, but I also take care of all the submissions from all the patients that are in any other place and want to participate in the study. So my work in the clinic starts identifying patients that are eligible to the study. I select the patients that have minimal change disease, FSGS, or membranous nephropathy, but there are also other patients that we don't know exactly the cause of their nephrotic syndrome yet. All these patients are eligible, and then I come to visit them in the clinic. Either their provider or me introduce the study to them, and if after reading some information and hearing what the study is about they're interested, then I would go to the next steps with them. The biosamples can be a saliva tube that we recollect either here in the hospital or we can send to the patient's address. We can also send a buccal recollection kit that is just a swab. We can also recollect blood, and that is when the patient comes the next time to the hospital. And we can also recollect something like urine or skin biopsies and kidney tissue if possible.
My experience with the Sampson lab this summer has been so great. I have loved my time here, and I have learned so much, not just about kidneys and genomics and stats, but I've also learned a lot about what it takes to be a good researcher and how to have a good work environment. And I found that when you're surrounded by people that are passionate, you also become passionate as a result, and everyone shows up and is excited to come to work and really try and make an impact on what they care about. So my biggest takeaway from this summer is that it really is okay to ask for help. I asked for a lot of help this summer, but no one ever made me feel like I wasn't smart or like I was wrong for asking help. And it honestly made my experience so much better. Everyone here has been so great.
Understanding the genetic basis of nephrotic syndrome is a big task. And that's not to mention, once we figure out the genetic basis of nephrotic syndrome, figuring out how to use that information to develop treatments, more accurate prognoses, and ultimately cures for the disease. It's such a big and complicated task that there's no way that one person or one group of individuals with the same expertise and background will be able to solve it all. So the value of having people with diverse expertise in our lab is that we all get to contribute something that we are excellent at together to create a whole that is able to address the different aspects of genomic discovery to implementation in nephrotic syndrome. For example, we have folks in our group who are experts in clinical care, some who are experts in the biology of glomerular disease, others who are statisticians, computer scientists, and folks who are experts in communication, community outreach. And all together, we are more than the sum of our parts, and that's benefiting our research and ultimately going to be able to benefit the health of our patients. And the last thing I'll say is, everything I just talked about was in terms of diversity of expertise in helping to discover and move research forward. But all of the people in our group are also of diverse backgrounds. They are raised in different parts of the country or even in other countries. They're of different sexes, different ethnicities, and that creates a wonderful mix of individuals who have their own perspective, their own stories that they bring together in our group to help us accomplish our goals in a way that is more effective, I think, than if we were all from the same background.